So, hey, everyone, my name is Dimitri Mills. Most of you guys have met me before in previous meetings. I'm here today representing a company called Uncurable Technology. Pretty exciting tech. These guys have come up with a way to identify and treat and cure certain diseases. And with the right team and forward momentum, we can add additional diseases to this particular platform over the course of the next coming years and change medical healthcare and things as we know it today. So let me, going to show you a couple of things. I'll show you one video. It's just a 2-minute video.
Yep, that's a big introduction, though. You have a lot to live up to there.
Well, I believe in the tech, man.
All right.
Joe, what I'm going to do is I'm going to.
Well, I think what he's saying is he's going to shut off his own audio. And
I'm sorry, Joe. What I'm saying is I'm going to come in on my computer so I can show you the other file. Can you accept my other instance?
Sure. As soon as you show up.
All right, we'll do.
Yeah. Andre, looking good? Let them hear your voice. We haven't heard from you in a while.
Oh, sorry, I'm late today.
I miss when Andre used to put the different picture in the background every week.
Very good.
He's recovering. He got really sick there for a while. Am I right?
Yeah.
Was it COVID related?
No, actually not. It was pneumonia, unexpectedly, but everybody thought it was COVID.
Oh.
I thought we got rid of pneumonia.
Nope, at least not in my case.
No. Sorry it was so tough for you.
Yeah, but I'm back.
All right. Okay, Jody has, okay, well, this is the second Dimitri, I think.
That's actually me. That's the real me. That's Dimitri. But I'm gonna play this video real quick, guys.
Okay, we're good. Ready to see it. Is there sound?
Yes, there should be sound.
Hold on. I'm sorry. Let me connect audio.
Okay, I hear sound, I think. No, I don't. And if this is on YouTube, you can even put on the captions so we can read it in case. This story follows the progress of 6 patients through their 12 days of treatment. HIVEX EMF treatment consists of an electromagnetic field tuned to disrupt the progression of the HIV virus. Okay, that's not working, I suspect, because you have 2 instances.
Yeah, team, this is Murto. Dimitri, go ahead and shut that down because this has to be tuned, let's call it, and coordinated properly for it to run over Zoom. That was done actually in the presentation that once Jody joins us, you'll be able to see and be able to hear it. It was tweaked for that reason because you're going video to video conferencing, so you have to adjust for that. And he just,
if you like, Murto, you can voice over. I mean, it won't be identical, but you could say what they're saying here is.
Well, no, I think the purpose behind this video was to give you guys the input from the gentleman that you saw right there with the CEO. His name is Steve Bosford. And we want you to get that impact, get that presentation. I wouldn't do it justice per se.
Okay.
So if we could just ask your patients a couple more minutes and he'll be on and we can roll.
Here with a very friendly audience.
We appreciate it. Thank you so much for the understanding.
So let me understand, neither you nor Jody nor Dimitri lead the company that we are being introduced to today. Is that right?
No, that's not right. So Jody Statari is president of the company.
Okay.
And the reason he will be giving the presentation is because he has the authority of history behind it, of course. And we felt that that was better communications to the team.
Okay.
Dimitri and I do have knowledge and can give you information regarding the company, and we're helping them to move this forward. But again, we don't want to mess up the presentation if at all possible.
I suspect that Dimitri Mills is Jody and Jody is Dimitri. So I'm going to.
I'm sorry, I apologize for being.
It's all good. I'm changing your name.
Okay.
S-T-A-R.
T-A-R-I.
S-T-A.
R. T-A-R-I.
Okay, got it.
Thank you so much.
Of course. And just one more thing, Murto, because you confused me there.
Yeah.
I thought you said the video showed the CEO.
That's correct.
Okay, and the CEO is not Jody. Jody is the president.
That's
Yeah,
correct.
Steve Botsford is the CEO. If I get into the presentation, then I can
Makes sense.
explain who's doing what.
So longtime fans of the show know that at this point, I'll say something that sounds a little introductory so that when I edit this later, it's not like, what? So, ladies and gentlemen, Jody Statari is president of Anapol Technologies. Jody, please introduce yourself and tell us about what you're working on.
Good morning. As you said, I'm president of Uncurable Technologies, and I'd like to talk to you today about, and hopefully you can see this.
Yes?
Great. I'd like to talk to you today about a emerging technology that's going to change the way that medicine is practiced. And we termed it resonance generating fields. I'm going to show you a short video that will give you a perspective of a practical application. It's basically hosted by Steve Botsford, our CEO, where we treated some patients in South Africa with the 5th generation device.
Okay.
We traveled to South Africa specifically because there is a clinic in downtown Durban that has been treating about 1,000 people over the last 15 months. The machine is an electromagnetic device that has a resonant frequency that is measured, and the people come in and sit around it, and they receive 2.5-hour treatment over 12 out of 13 days. The 8 people that had the most advanced stages of the infection, they typically would come in wheelchairs or walkers or being helped by somebody. Their energy level was completely down. Most of them would lay the first day or two either on the floor or on a chair. Very little talking, lots of despair. It was very depressing. But based upon the stories that I had heard from people that I trust, I was sure that over the next 10 days, I would see a dramatic change. The exciting part was the change started happening on day 3. You could feel the enthusiasm of the people. Everybody had a little more bounce in their step. There was a sense of relief. There was a sense of excitement. You're not thinking about dying right now?
I'm not anymore.
There was a sense of maybe we can get our life back that what we once had.
You know, for the first time I saw her walking by herself, it was like a dream to me.
And the only thing that had really changed is sitting in this room with that machine running that is specifically dialed in to the HIV infection. What I would hope is that by the scientific community learning more about this technology and the treatment that it does, that they would get behind it to be able to help develop it so that there's more things that it can do for mankind than just work one clinic in South Africa.
Without hope, you cannot get what you want, and with hope, you can be well again.
Amazing doesn't tell the story, but maybe that's the best word I can think of.
Well, that basically gave you a description of how it works in practice. And how many of you have known of someone, friend or family member, a relative, loved one, that's basically passed to a deadly disease, and while they were trying to extend their life, had to endure very, very difficult side effects? What would a safe, affordable, non-invasive therapy without toxic side effects be worth? So after years of development and testing, we're ready to commercialize our patented technology. In order to make this happen, we need a dedicated investment and strategic partners and alliances. We are not medically based, so we particularly like to interact and work with experts in the field. of each particular disease. The problem is that certain types of cancers and infectious disease abnormalities have no cure. They require, in many cases, lifelong drugs. They're very costly. They have some debilitating side effects, and it takes years to develop an effective treatment. We have an affordable, non-invasive, safe technology that targets disease progressors. And as you've seen from the video, patients sit in a gentle field. There's no drugs required. There's a low cost of treatment, no toxic side effects. And because it's a device and there's no metabolic effect of the field on the body, it would take months as opposed to years to develop an effective treatment. Now, how does it work? I want to direct your attention to the pictures on the right-hand side first before I get into more details. The top device is our 6th generation prototype. The device you saw in the video is 5th generation. The advantages of this device is that it's digital. It has a detection capability or diagnostic capability, and it's essentially made in America. The other device was foreign processed. So as you've seen in the video, the patient sits in the field. And the way this works is we can take specific targets, amplify their resonant frequency, transmit it into the field, and expose the patient to that. Now, how do we do this? Each target, let's say a specific microRNA in a disease is mutated. We will take that particular mutation and we will capture the resonant frequency of that mutation and amplify it and modulate the signal and transmit it into the field. And when the patient is exposed to an appropriately tuned field, what happens is resonance is induced in the target, which might be that microRNA, protein, the DNA, whatever is selected by the medical experts. And what happens is it significantly reduces the disease's ability to spread, and it drops down the disease viability. And these same resonances can be used to scan people to see the presence of the disease.
Jody, I'll ask you to stay on that slide.
Okay, just a second.
I suspect that more than one of us in the room have some questions at this point.
Sure.
Let me open it to the room. Does anyone want to start? David? David is on mute, but he is saying something very, very smart.
Okay, great.
Thank you. Thank you. It's amazing technology. So right when you said you're tuning it in, where are the people when you're tuning it to the right frequency? Because that could be a source of a hazard, right? As you're
right. As you noticed in South Africa, there was no Faraday chamber, right? And that's because we weren't targeting anything that's specifically produced in the body. All of the targets were part of the virus. With respect to cancer, there are some targets that can be affected and are produced by the body. So the way this works with cancer is we perform the treatment inside a Faraday chamber.
Okay.
And before we start, it takes about 10 minutes for the equipment to tune. So once it's up and running, and we can tell from the spectra, because the spectra is monitored the entire time. The new device has a broadband range from 100 megahertz to 6 gigahertz. And that way we can cover all the frequencies, the resonant frequencies, and their harmonics. So does that explain?
I think it gets me close. It's amazing technology. And one other word you had in your slide was no toxic side effects. And of course, when I see stuff like that, I always say, well, what did you look at, to determine that? Did you just?
Right. Well, in the trials, they basically compared all the bodily functions. We can send you the clinical trial reports.
Yeah.
But the kind of side effects that the patients experienced were some patients got a headache during treatment. Some patients fell asleep, and other patients felt a tingling, slight tingling.
Yeah. Did you measure anything though? I mean, was there anything you measured? And if this is on CT.gov, I can find it on clinicaltrials.gov if you registered it there. And
Yeah, these were international trials, but like I said, I could send you the
sure,
clinical trial report.
Yeah.
And yeah, there were all sorts of analyses on the patients in the control groups.
yeah.
So there was no real change over a period of I think it was 18 to 24 months of monitoring.
Okay, cool. Thank you, Joe.
Sure.
David, I'm surprised you said that you would look on clinicaltrials.gov and not just call it up on Nyquist data.
Well, I mean, that accesses it. That's what I was going to do. So, yeah.
Alyssa.
So, like David, I think this is a fantastic technology. The first things that I thought of was like, oh no, here comes another hokey pokey kind of thing.
Yeah,
but
exactly.
I think it makes sense if we can do MRIs and we can see every single particulate and ligament structure, and or not particulate, but ligament structure in the body. We can use an electromagnetic field to capture and target particles. However,
Right.
from a marketing standpoint, I would very, very, very firmly stay away from no side effects or no toxicities or anything like that, because as we know through the years, that's going to raise a red flag, and people are going to hone in on that.
Yeah.
So, and then that once they hone in on that, you're going to lose them for the rest of the presentation. So what I would say is, thus far, it's a safe, maybe we know that it's as safe as, or it's there's less electromagnetic radiation or field transmitted.
Yeah, that's a good point. It's transmitted at 0.001 volts per meter.
Yeah.
And the general population exposure is 1 volt per meter, or 10 volts per meter. I'm sorry. So very, very low.
So there you go. You can say, with comparison to that, this is the amount of electromagnetic radiation that the patient is receiving. So we are, explain how you come to that, but definitely the FDA will go, boom, I'm on it. If anybody says safe or not toxic, because you don't know how many people are going to, and how much time these people are going to be spending in front of it over time.
Right, makes sense.
Like Alyssa, I'm not going to comment here, but I too have some thoughts about how perhaps potentially to better tell your story, because the way you started, I was flooded with questions that made it difficult for me to even listen to what you were saying, because I wasn't caught up yet. I'm like, what am I looking at? But we
So
could do
you
that
think
another
a different order with the video?
Well, perhaps, the thing that struck me most was when the video was over, I felt warmth for your founder. That was the overriding feeling that I got. And I was touched by his compassion. And you went right into whatever slide number 2 was. There was no real acknowledgement of what human interaction we had. I also thought we really need to have subtitles on that video, because some of the speakers were difficult to understand at the volume that I was listening. Again, these are little things, and perhaps even before the video, instead of just saying, hey, watch this video, you'll understand, set it up with a sentence or two that is your value proposition. To whom, what's the primary benefit, and why should we believe? We can do this later on if it's valuable for you. Now, Alyssa and I have a shared marketing background. She's a masterful saleswoman as well. Mehdi is also a marketing person, so you're getting triple barrels on telling the story here. Mehdi, what have you got to contribute?
Thank you. Yeah, around the same lines. I think being more specific helps. One question I have is, what is the specific condition or body function or dysfunction that this corrects, and how? And the first thing that came into my mind was shockwave therapy, which is radio frequency therapy used in rehab a lot. It increases blood flow, it increases cellular activity, so it helps healing. So I wanted to know, how does it work? Is it a specific condition, and is this similar to anything that already exists?
It's similar but not exact. There's a direct competitor for glioblastoma treatment, which is what we're going to talk about in a little bit, that uses a backpack and a skull cap that's worn 18 hours a day to use electric field frequencies, and this is electromagnetic field. They basically suppress the magnetic component, but it's similar and it prevents cell division. In this particular case, the way this works is the equipment is not specific to any disease. So we go to an expert, for example, in HIV, the targets were ENV and GAG proteins, the viral envelope, and we basically targeted those proteins, some viral RNA and some pro-viral DNA in a suppression mode. So if we want to change from HIV, for example, to glioblastoma, in glioblastoma, there's a mutated microRNA that essentially is anti-apoptotic. So that was one of 4 targets that we used. When we change the target, the same device is applicable to the disease that's selected. So a very important portion of the treatment is getting the right experts to explain to us which targets to use. And then you can purchase the targets. They can be amplified by PCR, polymerase chain reaction. We get enough of a sample. We take the material, we stabilize it, and then we immerse it in an electromagnetic field that's modulated. We have different modulation types depending on the activity that we require. And that allows us to capture that frequency or a series of frequencies. And then that particular series of frequencies is transmitted and amplified. And the target itself is shielded, because what would happen if we didn't shield it is that the field would have an effect on the reference material, which is the target. So if you can take a look at that schematic to the right, you see where it says the stabilized target. That's where we pick up the resonant frequencies by essentially exposing it to the modulated field, and then it's amplified and broadcast in the room. And what it does is it forms an omnidirectional static-like field, and the modulation applies a torque to the molecule. And I think that many of you have probably heard the Ella Fitzgerald Memorex commercial where she shatters a glass by hitting a resonant frequency. It's not much different than that. Over a period of time, with the proper frequency, you have an effect on the virus or the bacteria. Does that make sense?
Loud typing there. I thought I was on mute. Apologies. Michelle, I am sure you have something to say.
I do apologize, because not only did I get in late, I have been a bit distracted because she has been on a tear the whole time. So
that's okay. I was late too, so no problem.
But kind of coming back to one of Alyssa's first points about not saying a safe or some of those words, but even saying safer is something that that is also a claim, and comparative this to that technology also is going to have to be supported by data with the FDA.
Right. In 2002, I met with the FDA. I brought 3 South African doctors over, and we had discussions with the FDA and the Office of Human Resource Protection. And one of the things that they were really concerned about was the fact that we were treating 18 patients at the same time. Groups averaged from, I think, 16 to 22. But there are ways to effectively control that here in the States. Mm-hmm. But there is, the safety was proven during these clinical trials in the optimum dose. So I don't.
Since you started your conversations in 2002, there's several new pathways with the FDA to facilitate clearance of new and novel devices, like Breakthrough and Safer Technologies Program. Yeah. This seems like this should be eligible for one of those.
Yeah, I agree. We just wanted to get to the point where it wasn't quite the prototype stage. And the old equipment, the 5th generation equipment, is 20 years old. And it's really not, it's not good enough to function properly now. So that's why we Americanized it and went digital. And this unit has been tested in glioblastomas, had similar results in cell studies, but not enough to show a statistical significance. Where with the 5th generation, we did show statistical significance with glioblastoma in cell studies, and we did have some animal studies that showed us tumor size reduction.
Jody, one thing that occurs to me is how many hands are going up, how many questions at different angles, and I believe this does not bode well, because we are all like, what about this? What about that? What about this and that? And if you are presenting this to, excuse me, sir, ma'am, can I have your money? This sounds like a really risky thing. If there are this many questions, we need something that's like, yes, yes, wow, oh my God, this is the new, holy shit, Beth, can I have the checkbook?
Right.
And I'm nervous for you already because I'm not hearing that. I have 2 more questions before I get to Ed and Paul. One is, you mentioned 2002. How long has this project been worked on? How many team members have been part of it at one time or another? How much money has been invested so far? And is it all friends and family at this point? If you could share some of that. I'm sure it's in the deck somewhere, but people have questions and they ask out of order.
Yeah, no, that's okay. Total investment's been around 17 million, most of which was spent in South Africa on clinical trials. And what happened there was the results were very good. And the South African government just unilaterally made a decision to go with the antiretrovirals. And that was it. So we decided we're going to basically bring it to the United States. And in the States, we've expended around $2.4 million, first starting with tuberculosis cell studies, University of Iowa. And we got a 2-log reduction in cell studies in H37RA tuberculosis. And following that, we ran out of cash for a while. And then back in 2011, we decided to see if we could bring HIV to the United States. We went all over the country and talked to various experts, universities, and they said, you can't bring this to use on HIV because HIV has a 95% successful protocol. You'll never break in. So the advice to us was go get something that's not curable. And we looked at 7 diseases, and the one that we thought was appropriate was glioblastoma for a couple of reasons. So glioblastoma was one of the first ones that was approved. The NovaCure technology, Optune, is the one with the backpack and the skull cap. And that's the first non-drug surgical therapy. So since we believe that we're a lot more effective and we can do with the 2-week treatment the same thing that they have to do with wearing it 18 hours a day, we thought that was the best way to start. And what can happen with this electromagnetic field, I can show you some other data. We can easily penetrate the blood-brain barrier, which is a problem for drugs. It's even a problem for the Optune system because the electric field won't go through, and they're transmitting with dipole antennas. And
we're now 4 questions deep after what have already been 3 or 4 questions. Again, I'm concerned. Paul?
Yeah, thank you, Joe. Jody, good to meet you. We connected on LinkedIn.
Yeah.
Apologies for the group, but what do you want to get out of this? This is a very powerful group of people here that have a lot to say and have a lot of very deep insight into the medical device, the market, bringing products to life. Yeah. Just so that we don't miss it, what are your top 1, 2, or 3 things that you'd like to get out of this meeting with all of us today, so we can at least frame some of our questions and maybe bring you in another time? But how can we help?
Well, we've been working with a lot of potential strategic partners, like Philips, Stanford, things of this nature. And what we have to do is to find someone that will have facilities in place so that we can reduce costs on the startup. So if they have the laboratory facilities, and they have essentially room for us to put in Faraday cage arrays. Because what had happened to us initially is with a single Faraday cage, everything has to be done sequentially. So something that would take 3 weeks now takes 12 weeks or 16 weeks just to get statistical analysis on one particular target. So we need to have a Faraday cage array with the various types of equipment, and that's going to cost money. And if we can establish it with a facility that has the laboratories, that can do analytics, things of that nature, that'll make things go very fast. And of course, we need the funding. And
you need more funding after Dimitri? I thought Dimitri had all the pockets. Dimitri has all the pockets you need, right?
Yeah, exactly. He's got all the emotional cash anyway.
So funding. By the way, have you before I sorry to cut you off, but have you all considered, are you open to joining medical device accelerators, or medtech accelerators? Have you done that yet? Because they can bring you closer to financing, and they might have an infrastructure.
Dimitri, you want to talk about that? Because I'm not exactly sure whether we're tied into that or not.
Yeah, Paul, we'll take a look at that, and we can talk about that offline, Paul, and see how we can move them into those areas as
Joe's
well.
got some connections in that space. I'm following Joe, and it's a very close knit community. My next call is with the CEO of MedTech Innovator at 11:15 today. So, but Joe's got all this contact.
Name drop her.
Brazen Bio.
Yeah, go ahead.
Yeah, Brazen Bio, the founder of Brazen Bio, developed a glioblastoma treatment, and I think that's an accelerator that would be, I think, right in line for you guys.
Interesting. Thank you.
And the third thing you were going to talk about before I cut you off, Jody?
Well, and we realize we have to go all the way through phase 3 trials. So that's going to take a lot of money. We're basically looking for initial ask of 11.2, which would get us through glioblastoma feasibility. And then totally, if we look at both markets and you haven't seen this, a glioblastoma, and the second one that we want to target is triple negative breast cancer. We have some relationships with experts in the field. We do have some limited treatment experience in breast cancer. Those two markets would easily allow us to reach $1 billion in sales in about 5 years, and that's at about 65% of the median price that's charged now. Wow. So we that's what we need. We need medical partners with facilities on site. We need the money, and we need to fix the minor. We need to optimize the prototype. We just haven't tested it enough. And it's not like the old technology was lost, because the targets that were used in South Africa, for example, the GAG proteins, the DMD proteins for HIV, are compatible with the equipment right now. So we could basically do that if the strain were South African HIV.
Eddie?
Hey there. First and foremost, by full disclosure, I'm not a virologist. I'm not a doctor. I'm not a bacteriologist, but I'm fascinated and intrigued by what you're developing. And couple of questions. Let me just rattle them off. Number one, have you spoken to virologists or anti-infective folks about the mechanism of action and why it works? Then my next set of questions is, I have some other background, and I understand why and how you might be able to argue that what you're talking about in targeting these viral proteins or these other structures of these other, if you will, organisms or semi-organisms differentiates you and keeps you separate from the human cells. But I'm intrigued as to why you've shifted from virology effectively to cancer, to oncology. Because when you start describing, we're going to attack these cancer cells. Well, cancer cells are human cells that have just gone wrong. So it raises, to my mind, a lot of issues of safety that
and
maybe you got the data on, I don't know.
Last
but not least,
okay.
I know I'm throwing a lot at you. Last but not least, although you're a device, you also seem to be a therapeutic. So again, from a regulatory standpoint, I don't know how you regulate it, because you're talking about dosages. And when do you know you got a therapeutic dosage? How, do you have to stand close enough to the machine or what?
It's basically related to the exposure time, and each particular disease is different. And the way that we can see an effect with respect to the equipment, not necessarily the patient, is that there will be a shift in the spectrum over a period of time. So like if you look at the spectrum on day 1 and then you compare it to the spectrum on day 3, there'll be a shift in different areas where there are resonance. And after, for example, with respect to HIV, after 30 hours of cumulative exposure, there's no change in the resonance. So we know that whatever effect is going to be applied has been applied. And that's how we can tell whenever we're operating. With respect to the various types of cells, and as you said, these are just human cells that have gone bad. So the best approach for us is to go as far down the chain as we can. In other words, like I had said before, with glioblastoma, one of the targets that we used was this mutated microRNA, I think it's MIR 381. So that's only available in the cancer cells. And you say, well, it's made out of the same substance, right? It's got hydrogen, it's got carbon. But a very simple principle is that if you've seen people that use wine glasses with various levels of water, and they rub the top of the glass and they get a different frequency. Same thing applies.
Yeah, I've seen miscongeniality. I understand.
Yeah, yeah. But that's the principle, right? So we have done a lot of studies where, for example, when we did the tuberculosis studies at the University of Iowa, they actually harvested human macrophages to go and be infected with the tuberculosis. So the macrophages were exposed to the field along with TB. And after the period of exposure, there was no difference at all in the macrophages that weren't infected. So there's a lot of data there that shows it's very, very specific. But again, when we did do cancer, like another target for the glioblastoma is HP70, which is a protein that's anti-apoptotic, and it's overexpressed in glioblastoma. So there is some effect that's going to happen to the normal HP70 in the body, which is why we said, okay, we're doing cancer, we've got to shield it. And then shielding gave us a few problems because of reflections and stuff like that. We've resolved that. Did that answer your question, Ed?
Yeah, and that answers my question pretty darn well. Like I say, I think if you've got, I presume you've got, or are you looking for access to doctors who actually can speak with authority on these sorts of things, as I certainly can.
Yeah.
Andre?
Oh, yes, thanks, Joe. Hi, Jody. Just like Ed, full disclosure, I'm not in the medical side, but actually my area of expertise is in EMF. We have a wide number of patents in EMF non-invasive therapeutics and have gotten 3 different versions put through the FDA and also with international approvals and so forth.
Yeah.
We've done a lot of studies on these signals, on certain signals that have cellular effects and so forth. The end result, of course, is whether or not you can, get the regulatory approval. And hearing all the things that you're going after, the only concern I have is that it's such a broad base that it might raise some red flags. We've learned over the years, we've been doing this for a number of years, that focusing on a specific indication with a specific modality is probably your best route, number 1, to funding, and then number 2, through the FDA, and then you could bring on the other indications. Of course, with all the safety studies and so forth. But we're on the device side. We develop all these products. We also are a public company, and when we've developed these products and patented them, we've then spun them off into other companies in order to get them funded individually. So, I'm very interested in hearing what you're saying and what frequencies that you use and so forth. Obviously, we've looked at the full frequency spectrum and what the different frequencies will do in the body. And we have both in vitro and in vivo analysis of the cellular effects of EMF therapeutics.
Right.
So very, very interested if I could connect with you. Joe always talks about.
Yeah, that would be great. One of the things that we think is unique to us is the antenna configuration. We don't have dipole antennas. We have multi-solenoid antennas, which gives us a unique field. And it forms, as I think I said before, a static-like field, which the modulation basically exerts a torque on a particular target. And we used to think that it was completely destroying it. Now we're not sure. We think it may shift the frequency just enough so that it can't hook up, right? It just deflects the receptors, things of that nature. We don't exactly know. In HIV, what happened is the patients got better. The immune system was able to recognize the virus. It wasn't running away. But if they went out and they got infected with a fresh strain, they're not immune. And it would put them right back in the soup. So the particular mechanism of action with HIV, we don't know for sure.
And Jody, I've been in the back of my mind, I've been worrying about the whole idea of, because it seems like you've leapt straight into human trials. And that poses a huge amount of other risks, particularly because you can't do controlled trial. I mean, the nice thing about it is it seems like you could easily do a placebo-controlled study, but there's lots of ethics about doing that. Have you got any animal data? Because there are loads of animal models.
We do, yeah. We have animal data from the clinical trials in South Africa. They basically exposed, I think, 4 generations of rats. I mean, that's published. When we did the animal studies in Albany was where we have doing glioblastoma studies. We exposed the mice to over, I think, 600 hours of continuous exposure with no negative side effects. And in each of the studies we had with the mice that were, these are flank-induced injected mice with glioblastoma cells. They weren't into the brain, but we were just starting. We had 5 sentinel mice in each of the groups of 30 injected mice, and none of the sentinel mice had any side effects at all. So there's a lot of data out there that shows there's not an effect. This is anecdotal, but I've been in the field for, I don't know, probably 3 years cumulative time. Other than losing my hair, I don't think there's anything else that happened. But yeah, I don't mean to make light of it, but there's a lot of data out there that shows that it's very specific, and it's not much different than an MRI that way.
Jody, do you intend to pursue reimbursement in the States for treatment?
Yeah, yes.
Do you want to come off mute, Mr. Anderson? Nick is the go-to guy for all matters reimbursement in the United States.
Yeah.
And many other things. Are the twins born yet? When are they coming?
Thanksgiving.
Oh, okay. I'm ahead of myself.
Congratulations.
Thank you.
He wanted 6 children, so this was the quickest way.
We tried to have all 6 at once, and that just didn't work.
Have you heard anything reimbursable in today's presentation?
No, there'd be quite a bit of work to do. I like the comment of what Andre said. It's oftentimes what I refer to as putting the camel's nose under the tent. Find one indication with one narrow, ridiculously narrow population that your board of directors will hate you for because it's just so narrow. There's 5 people in the galaxy that have it. And you go get reimbursement there and you say, all we're asking for is a medical policy from Aetna for glioblastoma multiform between the ages of 20 and 25 that don't have diabetes, some ridiculously narrow thing.
Oh, interesting.
Recruit those patients, nail it, home run. We proved that we could do it, whether it was the diagnosis or the treatment or whatever, again, to Andre's point, is it diagnosis or is it treatment? So if you say, we're just going to do diagnosis and it's so narrow, again, your board of directors will think you're crazy because they're under the impression this is a $1 billion market, but no company on earth, Amazon doesn't have enough money to prove a $1 billion market in medicine. You prove this much of the camel's nose, you get that much under the tent, then you do 25 to 30-year-olds and then 30 to 40, 40 to 90. And then you go on to the next indication and you rinse and repeat until that medical policy at Aetna or Cigna or Medicare is just growing. And they say, man, this company is doing it right. They're proving this, we give them reimbursement. Then 3 years later, they prove this and we give them reimbursement, and you rinse and repeat until all GBM patients are covered. And then you go on to do uterine fibroids, or you go on to do glioblastoma or macular degeneration or something. And you just keep shoving more of the camel's nose under. But if you try to eat the whole thing at once, That's a $500 million study, which doesn't exist. No one's ever done a $500 million study that I know of. And it will take 40 years to do. Your bell curve, your confidence interval is going to be so washed out with noise that the insurance policy will say, due to the heterogeneity of the evidence, we cannot ascertain the clinical benefit of this technology. And it is by consequence investigational, experimental, and not medically necessary. That term, due to the heterogeneity of the evidence, is exactly what Andre just said. That Aetna, Cigna, Medicare, and all of them will say, your evidence is too heterogeneous. We cannot tell, even if we did a subgroup analysis, which we at Aetna are not in the business of doing biostatistics. That's your job as a medical device company. It's a hodgepodge. You had 20-year-olds, 80-year-olds, you had patients with stage 1, stage 4. It's a hodgepodge, and we're going to say, due to the heterogeneity of the evidence, we ain't paying for this.
Yeah, that makes a lot of sense, and it does fit with. I didn't show you the path forward, but what we are doing is looking at cells studies and then the animal studies. And when we did the glioblastoma, it was specific to U87MG, which is a specific strain of it. And looking at a 10 to 12 patient feasibility study would have essentially been on that particular strain. So, you know, if you look at the capacity ramp up, what you're saying is not much different than what we had planned. It just doesn't come, you know, I'm not verbalizing it that way, but that makes a lot of sense to me, what you're saying.
Just as a note, there were another 3 or 4 comments that we didn't get to, and you got up to slide 5. This is a problem.
Yeah.
This is a problem. I'm curious about what the other slide said. We are at the top of the hour. I am recording. Sometimes people drop off. They can watch the balance. How many slides are in the deck?
There are a total of 14. We're on slide 6.
Okay. Well, if you would, you may have noticed I took off the screen sharing so we could all see one another a bit better. If you could just like really skate through them, we won't comment. I just want to see what the rest of the story would have been.
Okay, no problem. Really quickly.
Yes, please. Okay.
Okay, we just talk about advantages and you've seen it. Yeah.
If I may, yep, next slide. Primary targets.
Yeah.
Okay, next slide.
Yeah. This is why we're doing it.
The page, actually, the one that you skipped over fast is the one I'm curious about what you're saying. You were saying you had 2 markets that you identified?
Right. That's to start, yeah. The glioblastoma, since we've done all the work on it. And then triple negative breast cancer, we have a very good relationship with an expert in the field, 25 years on it.
Thank you.
Yeah. So I think it's a good other place for us. This is why, and you know, things are changing that accept the technology now. It's very hard before to get in. This is the value generation. I mean, people that get glioblastoma, 14-month median is a death sentence. Only 7% made 5 years.
This is an unfamiliar format, so I'm not really following the story easily. What does this, without looking at numbers, what does this slide say? Are you trying to say this is how much money will be saved in the value chain?
No, the value chain is what the estimated sales are and the difference between the conventional treatment cost and what we're showing is a break-even cost. Not at capacity, but a very, very limited capacity, 22K a month. And if we look,
I'm not alone when I say I'm still not really getting the point. I think you're saying that you need to make $22,000 in sales to recoup your investment in the device.
No, I see where you're going. At 30, if we take 50% of the new cases or 16% of the glioblastoma active cases in the U.S. alone.
Mm-hmm.
And we basically have a price of treatment at 65% of what the conventional median cost of treatment is. Okay. That generates 35 million a month with just 600 patients a month.
Okay.
And it generates a positive cash flow of $21 million.
if I were the investor, I'd be like, where did all 16%, where did all these numbers come from? That's rhetorical. That's
no,
what
it's not rhetorical. It's based on
no, my comment is rhetorical. I'm not asking you to address it here. I'm just like,
okay.
what?
Yeah.
Next slide, please.
Yeah. These are people that we've been in discussions with about being strategic partners and helping us ramp up.
Okay.
This is a team.
Very, very handsome team.
Thank you. And this is the path forward. And I think it was Nick that was talking. Is you can see here we have proof of principle cell studies. I would expect it would still be the UA7MG go through, and only 10 to 12 patients, or that could be made to be very narrow, which is probably what's going to happen. And as you said, once it's proven for one, then you basically go to the next step.
I'm thinking that the timeline is pure conjecture because it's all a function of money that you don't have.
Right.
So why don't we put together a, T+1 thing, once we get the money, then we can start this timeline perhaps, because we're already in the third quarter. Do you have $200,000 for these 4 months?
Yeah, that's been funded, it's self-funded.
Okay.
That's been going on for the past more than 4 months.
And the next 2.1 million also self-funded? That happens in 2 months?
No, that's not self-funded.
Right.
So what you're suggesting is a plus.
It just,
+1. Yeah, that makes sense.
If we were to get this far, because I don't typically sit in VC meetings, but I mean, they would have to be silent the whole time to let you get through all 14 of these slides.
Right.
And at this point, they've shot so many holes in it for you to say, so don't worry, by 2026, we're going to be in the money. Dun,
Yeah.
dun, 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 dun. That's about what? Sorry, I went all Bugs Bunny on you there.
No, but I think that the feedback is very helpful. And
Well,
you're actually the first people that have seen this.
Oh, okay.
Yeah, so.
Well, Dimitri did start by saying that you guys have spent quite a bit of time to prepare for today.
Yeah.
I didn't realize that. So if I understand you, then is it just this iteration of the deck, or have you not made a presentation like this before investors before?
We've made a presentation, but not this particular deck. And it's gone back and forth. There's so many questions. What happens is we have a short discussion and then come back with questions and we have another discussion.
Okay. Murto, I'd like you to come off mute, and I know you came on camera for just a little while there. You want to add anything today?
Yeah, first of all, thank you to the panel for your time and your suggestions and comments.
I know you're a good-looking guy. If you could put the camera back on.
You came on for a second. I'll send you a mugshot, but we appreciate the input. We really do. It's a refinement process. We get it. I think we've heard a lot of significant ideas and comments here that we can take with us and make some adjustments. We look forward to possible more communications with you folks. I saw that there was a couple of folks here that are interested in maybe doing that. I think with Andre was one of them, for example. And so just again, thank you for the time, and we appreciate it. We'll take these suggestions to heart, and we'll make some of the changes that you guys are recommending.
There were a couple of comments too. I'll make sure that Dimitri gets them. They were typed up. Folks, Dimitri is a fully fledged MDG premium member, so he is on the network. Andre, I've already sent you a link directly to his account, and a number of you have expressed an interest in following up with him directly. So David, respectfully, I'll not take your question here. I'll have you just follow up with Dimitri directly afterward. This was these types of meetings are the ones that I feel add the most value. We have some great talks and we're like, oh, that's really interesting. And then we go on our day. This one is you're going down a path and I get some really smart people in the room and say, did you think of this? And did you think of that? And I think that's where real value is. So thank you, smart people, for joining us.
Yes, thank you very much. Thank you all. It's been an eye-opener. We really appreciate it.
If you want more on reimbursement, Nick is our star performer next week, and he's always a good time. The week after that, I'll be on to tell you what all but Eddie missed by not being in London with us. So Eddie, you'll participate and keep me honest and say if it really was as good as I say.
Absolutely. And Joe, just so you know, I'm going to be in town the night before, and particularly because Thursday is yet another of our famous rail.
I heard that's a effing
So
risk.
the entire National Rail Service is canceling everything they're doing just so they can be with you on Thursday.
Let me understand, is that definite? Is it definitely
Yeah.
happening on the 15th?
It's happening.
This.
But
I don't know
B,
if any of you folks are religious at all, but what the fuck, God? Wasn't COVID enough to fuck me up as a conference producer for 2 years?
Look, it's the railways, but not the tube. And it's
Yeah,
not all the railways.
I get it. I had one guy
Trains
is
will
like,
still run.
I was ready to pay, and then I see that there might be a rail strike. And I'm like, oh, that's really? Because
You're doing something wrong, Joe.
I hope you're recording this, Joe.
I am recording this. I'm going to send it straight to God right after this. I'm
I
also
feel
going
like
to
we should
send
have
it-
a Joe pitch.
I'm also going to send this to the bursar at Boston University and explain why this year's payment is going to be shy 495 quid. Yeah, it's all funny until you pay the bill. Ladies and gentlemen, this is Joe Hage for Joe Hage Enterprises, growling at you. Join us next week, same bat time. Thanks, everybody.
Thank you.
Jody and team.
Thanks, Joe. Thanks, guys.
Okay.
Take care.
Bye, everyone.
Bye-bye.